welcome back to my channel by Mix Up. In this today's video, we are going to discuss about the enzyme that is DNA ligase. So where does this DNA ligase work? This DNA ligase work in the resealing of the DNA. So where does the DNA need to be resealed? Suppose there is a synthesis of a lagging strand. During the synthesis of the lagging strand, there are short primers which are being synthesized by the enzyme primase. After the synthesis of the primer, this primers needs to be removed. So after the removal of this primer, there will be a gap in this DNA. The gap will be resealed, or we can tell the gap will be synthesized by the DNA polymerases, and the last pair of the nucleotide will be resealed. After synthesis of the nucleotides, the DNA, two strands of the DNA, they needs to be resealed. So during this time, there is a breakage in the phosphodiester bond. What happens in the last? There is a breakage in the phosphodiester bond, due to which there is only one phosphate present over here, and there is a free 3' OH group which is present. But this process, the bond which is formed in this reaction is a phosphodiester bond, and in the phosphodiester bond there is requirement of energy. So from where does the energy comes? So for this, the enzyme which is present for this ligation reaction is a DNA ligase. So what is the first step of this DNA ligase? The first step is that it will help in the adenylation. We will be very clear with this help of this diagram, with this picture. The first enzyme which is required, the first thing which is required in the step 1 is adenylation of ligase. That is involvement of ligase for the ligation reaction. So this ligase enzyme which is present, it is having a NH3 group with it. Okay, so the first step is adenylation of ligase. So during this adenylation step, there is requirement of ATP, that means adenine triphosphate. Here we are having 3 phosphate, and for its adenylation of this ligase, 2 phosphate are being removed, and this only 1 phosphate is being added with this ligase enzyme. Okay, we can see here that this is now the ligase AMP. Ligase enzyme is present, along with it AMP is present. Okay, this is the ribose sugar, and this is the adenine. This is the adenine. So what is the second step? Here we can see that this is the Nick DNA, and in the Nick DNA, the 3' OH group is free over here, and due to break up the phosphodiester bond, here only 1 phosphate is present. So here that cannot be attacked with this OH cannot attack with this phosphate group for the formation of phosphodiester bond. So what happens in the next step? Here the ligase AMP is formed. This ligase AMP, what does it? It transfers this phosphate to this first phosphate. Okay, so here we can see that this was the original phosphate which was present beforehand, and this is the phosphate of the DNA ligase. This whole is the ligase AMP. This whole is the ligase. This is the ligase AMP. Okay, this is the ligase AMP. We can tell that this phosphate group of this ligase AMP is transferred to this first phosphate. Okay, so now what happens? There is a nucleophilic attack by the 3' OH group. So the 3' OH group which was present, which was free, it can now attack with this phosphate now. So after this attack of this phosphate for the formation of phosphodiester bond, it requires energy. So here this phosphate is being removed. After it is attacking to this phosphate group along with this AMP is removed by the bond formation, that is the phosphodiester bond. Okay, after the phosphodiester bond is being formed, now the DNA ligase has done its work. That means it has sealed the DNA. We can see here that after the formation of the phosphodiester bond, the DNA is sealed here, and the AMP is now released. This is the work of the DNA ligase enzyme, and this is a mechanism how this DNA ligase performs its work. Thank you for watching this video till the end, and if you like this video, please do like, share and subscribe. Thank you.